Today I will present about the theoretical characteristics of East Asian herbal medicine, and I will call it EAHM from now on. And I will present the necessity of introducing quality evaluation systems reflecting EAHM characteristics, because I think it will become increasingly important in the field of pharmacopuncture too. Herbal pharmacology is a study of the origin, quality, and pharmacological efficacy of EAHM. So, I am very delighted to present on a related topic as a professor of herbal pharmacology. And as I am the last speaker in the session, so I will make a quick short presentation. First, let me explain about the theoretical characteristics of EAHM. As you know, the biggest feature of EAHM is that it treats sick person instead of treating person sickness. To do this, syndrome is used as a primary parameter to choose appropriate EAHM instead of the symptoms. The syndrome can be defined as a collection of human phenotypes based on holistic unique classification theory. As shown in the figure, people with different disease names can be classified to have the same syndrome from the perspective of EAHM. Also, a person may be classified into multiple syndromes, and the syndromes can change as time goes by. EAHM administered for the appropriate syndrome at the right time is effective regardless of the disease names or individual symptoms. Therefore, it is necessary to reflect these concepts of syndromes and holistic perspectives on the quality evaluation system too. Advanced technology can also be applied to the deeper understanding of EAHM. As seen in the left blue boxes, extensive clinical information of EAHM can produce new insights and knowledge through big data analytics. And the holistic and unique perspective focused on the syndrome can be foundation for the phenotype-based approach and personalized approach of the modern medical science. And the unique principle of the drug combination can play a key role for achieving the synergy based on the multifaceted polypharmacology. Therefore, our team aims to study phenotype-optimized assay target and target-optimized experimental model and phenotype-target-optimized candidates. To achieve this, I am studying with the goal of building a standardized research program. This can be seen in 4 stages: multidisciplinary approach, integrative hypothesis, and decision making, and the actual achievement. First, I will briefly explain about stage M, which utilizes multidisciplinary information. Through SOP M1 to M4, all data on EAHM, including clinical research, preclinical research, classical literature, and industrial information are collected. And these data are systematically analyzed in a multidisciplinary way using big data and AI. In stage I, a hypothesis about the operation among the phenotype target compound is performed based on the analyzed data from stage M. And the hypothesis about the synergy in drug combinations of EAHM is also performed. These are some examples of most recent results using these M and I stages. A study which aimed to clarify EAHM target syndrome by analyzing the phenotype, target, and its network was published in Pharmacological Research last year. Research question of the paper was, can key drugs in herbal formulations targeting different syndromes and exert consistent therapeutic effects and mechanism against the rheumatoid? And we suggested the answer that phenotype target complex network to clarify EAHM target syndrome against rheumatoid. The second example is a study that explores the dimensions of the synergy in hub pair between EAHM. Next, in stage D, experimental model that reflects the syndrome is designed, and the efficacy of EAHM is evaluated using this in vitro and in vivo model and compare the effects by dosage and solvent and process, etc. Also, the multi-compound, multi-targeted mechanism of EAHM is identified through informatics and pharmacological approach to find the synergy. Next, quality evaluation is pursued based on the derived active compound. Furthermore, we aim to establish pharmacological marker that reflects the syndrome and standardize the EAHM with the efficacy. By scaling up, we are working to build a standard procedure so that it can be reflected in the end product. In this process, there was some achievement in patents, projects, and clinical or technological transfer. Today, I would like to talk a little more about one of our projects. The research subject about quality evaluation is currently being carried out. It seems that the current problem is to evaluate the quality of EAHM only with a single compound contents, as a second speaker said. Although the pharmacological effect of EAHM is pretty comprehensive and holistic. So this project is innovative because it aims to establish pharmacological assays that reflect the characteristics of EAHM. In short, EAHM has two characteristics. One is that is a natural product, and the other is that has been used traditionally. Considering that it is a natural product, a paradigm shift was needed to discover multi-compounds and multi-targets instead of the current magic blast paradigm. In addition, considering that EAHM has been traditionally used, it was necessary to set up an assay that well interprets the theoretical characteristics of EAHM. For this, it was necessary to focus on the syndrome again. I will explain about a bit more about this. We are researching some prescriptions, and I will take the case of Soshotang as an example. Because Soshotang can show many aspects of the some representative points, so second present show the same prescriptions as a representative example. Soshotang is, as you know, a representative prescription to treat Soyang syndrome. In other words, Soyang syndrome, represented in green color, is a state where the disease lies between the exterior, the yellow color, and the interior, the blue color. 소양 syndrome leads to various symptoms underlined, 한혈악내, 흉욕고만, 묵묵부려검식, etc. And 소조탕 is one of the most representative EAHM which treats 소양 syndrome. There are different expressions in Korea, China, and Japan about the indication of 소조탕. It looks very different. However, if you have understanding about the theoretical characteristic of EAHM, you will not see this superficial difference. You can see that 소조탕 is trying to treat 소양 syndrome that penetrates the whole. Indicators that can evaluate 소양 syndrome were collected through stage M and I using informatics technology. Then hypothesis was derived. Target assay was also derived, including immune-related inflammatory disease, fever, anesthesia, upper respiratory infection, chest side pain, etc. This result was also consistent with the ICT study of 소조탕 examined by ICD disease code. So our expectation was not that wrong. Our team is evaluating 소조탕 with collaborators through various in vitro and in vivo models, which was set in the stage D. We are also conducting research to explain the efficacy and the synergy of social tongue through an informatics approach. You can see right here. It's my last page. I think EAHM contains a lot of wisdom itself, but there is a big problem that quality evaluation is very, very difficult. It is a problem to simply evaluate the quality of EAHM only with a single compound content. It will be possible to improve quality control and create valuable evidence through informatics and pharmacological approach by reflecting the syndrome and holistic perspective of EAHM. It is a quite challenging task, and there are many things to be clarified, but it would be great if it could be reflected from a long-term perspective. And I believe the value of EAHM can be maximized if researchers and related parties cooperate. These are my researchers working together. Since there was no group photos, so I took this photo yesterday. Thank you for your attention on my presentation.